Hello, everyone, and welcome to today's webinar titled PCR Testing Reveals that SUC is Not So Golden of a Standard of Care. This webinar is a part of the 13th Annual Clinical Diagnostics and Research Virtual Event. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, please go to thermofisher.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You will also notice that the speakers have provided a survey located to the right of the Ask a Question box. This survey will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speakers, Becky Winslow, Clinical Science Liaison at Acutus, and Dr. Mayil Shanmugam, Senior Scientist at Acutus Diagnostics. For a complete biography on our speakers, please visit the Presenter tab from the menu at the left of your screen. Ms. Winslow and Dr. Shanmugam, you may now begin your presentation.
Hi, my name is Dr. Becky Winslow, and I and Dr. Mayil will be discussing with you today polymerase chain reaction testing and how it reveals the standard urine culture is not so golden of a standard of care. So let's jump right into the impacts of urinary tract infection, as they are quite extensive. Urinary tract infection affects approximately 150 million people worldwide. Urinary tract infection is also the most common outpatient bacterial infection in the U.S. Urinary tract infection leads to 2 to 3 million emergency department visits per year. It leads to approximately 10.5 million office visits per year. It causes 3.5 billion in healthcare related costs annually. And last but certainly not least, urinary tract infection leads to workforce presenteeism and absenteeism. So let's look at the standard urine culture and some of the technical challenges associated with the standard urine culture. True or false? The standard urine culture was validated for use in identifying lower urinary tract microorganisms. What do you think? False. The standard urine culture was not actually validated for use in identifying lower urinary tract microorganisms. Yet, for more than 60 years, the medical community has relied upon the standard urine culture for identifying uropathogens. The standard urine culture was never validated for use in identifying lower urinary tract infections like bladder or urethral infections. And those, cystitis in particular, represent the majority of urinary tract infections. So let's look at why the gold standard for identifying uropathogens is not so golden after all. The standard urine culture results in a high rate of false negatives. Why? Because the standard urine culture is biased to grow E. coli, And with standard culturing protocols in place, the standard urine culture will not detect slow-growing organisms, fastidious and non-aerobic organisms, and most gram-positive organisms. The standard urine culture also can misclassify polymicrobial infections as sample contamination. It can take the standard urine culture up to 72 hours for a turnaround time. And last but certainly not least, previous exposure to antibiotics or the patient's urine exposure to antibiotics can skew the culture's results. And the bottom line is that just because pathogens have not grown in standard urine culture over the last 60 years does not mean that they were present in samples. And this has led to undiagnosed and misdiagnosed urinary tract infections as well as antimicrobial resistance. So let's look at polymerase chain reaction and its advantages in urinary tract infection pathogen identification. The main take home that I want you to have from this presentation is that polymerase chain reaction is advantageous because pathogen detection and identification is not dependent on the organism's growth in culture. So let's take just a minute to look at a research study that compared PCR versus culture. In a retrospective record review of 582 consecutive elderly patients presenting with symptoms of lower urinary tract infection, all patients had traditional urine cultures, and they also had PCR molecular testing run in parallel. A uropathogen was detected by PCR in 56% of those patients. And by traditional urine culture, the uropathogen was only detected in 37% of patients. That's quite a significant difference when we're talking about clinical care of a patient. Now, the PCR and culture agreed in 74% of cases. Both 34% were positive and both 40% were negative. There was disagreement between PCR and culture, and that was 26%. The PCR was positive and the culture was negative in 22% of cases, and the PCR was negative and the culture was positive in 4% of cases. There are reasons why a culture may be positive versus the PCR being negative. That goes beyond the scope of this discussion, and we won't cover that today. We can cover that at a later time. In the 175 patients with polymicrobial infections, PCR detected 166 pathogens versus culture that detected 39 pathogens. So these results suggest that PCR may be likely to identify pathogens before traditional culture will. And this very last statistic is highly clinically significant in that polymicrobial infections were detected in 12% of the samples in which the culture had actually given a negative test result. So for 12% of those samples or 12% of those patients, they may have been not treated at all for a urinary tract infection when in fact they had a polymicrobial infection. So let's just take a look at the clinical journey a patient would experience in which a prescriber, prescribes standard urine culture versus PCR for diagnostic testing. The standard UTI diagnostic workup looks something like this. The patient presents with signs and symptoms of lower urinary tract infection. The doctor performs a urinalysis via dipstick in the office. Based on the urinary analysis results, the doctor may send the sample out to the lab for a standard urine culture. The doctor will prescribe an empiric antibiotic before the patient leaves the office. And based on the standard urine culture results, the doctor will call the patient in 3 to 4 days to deprescribe, adjust, or maintain the empiric antibiotic. So the only change in this workup procedure when a doctor decides to use PCR testing is that they send the patient's urine sample for PCR testing at a lab that provides PCR testing versus sending for standard urine culture only. So you can see that PCR testing is not disruptive to the standard UTI diagnostic workup. Now, very importantly, let's look at the timeline for the patient, for the prescriber who uses PCR versus standard urine culture to diagnose their urinary tract infection. So let's say that Lab A has PCR, urinary tract infection testing, and Lab B has standard urine culture testing only. Okay, on day 0, the sample arrives at the lab, no matter which lab you send it to. On day 1 at the PCR lab, the PCR ID is reported, a preliminary AST is reported. However, on day 1 at Lab B, the sample is only being plated and to run for culture. What happens on day 2? At Lab A, the AST may be completed depending on the pathogen, but at Lab B, you can see that the sample is still running for culture. On day 3, at Lab A with the PCR testing, AST is likely completed depending on the pathogen. But down on day 3 at Lab B, the sample is still running for culture and the results might not be done. At Lab B, you can see that on day 4, the sample is still running for culture and the results might not be done. On day 5 at Lab B, the sample is running for culture, the results might not be done. And then on Lab B, the ID and the standard urine culture guided AST report may or may not be complete. So what is day 1? How does day 1 compare for the PCR lab versus the non-PCR lab? There's a faster turnaround time to accurate identification of pathogens or the absence of pathogens. There's also a faster turnaround time to deprescribe or prescribe. How about day 2 or 3 for the PCR lab? The PCR-guided AST has accurately validated or invalidated the prescriber's original treatment selection. So this leads to a faster turnaround time to prescribe or deprescribe. Let's look at the standard urine culture day 4, 5, and 6. A slower turnaround time to less accurate identification of pathogens or absence of pathogens. We also have a slower turnaround time to prescribe or deprescribe. So you can see from that timeline that the PCR-guided urinary tract infection testing produces a faster turnaround time for results, and it also leads to a faster deprescribe or prescribe. So let's just look at a summary of how PCR UTI testing can interrupt the progress or the typical scenario, which is where standard urine cultures that are undiagnosed or misdiagnosed can lead to the negative clinical and economic outcomes. If we use PCR instead of standard urine culture for the identification of uropathogens, we're able to decrease the negative clinical and economic consequences that we see from the standard urine culture. So now I'm going to hand over the presentation to Dr. Mayil, who will discuss with you the rigorous scientific and regulatory validation that PCR testing must undergo. And I want to highlight before he starts how critical it is to clinical outcomes from urinary tract infections that these tests undergo this scientific and regulatory validation process that he is going to describe for you. Okay, Dr. Mayil.
Thank you, Dr. Winslow. So any assay we develop, it has to go through three processes. First, it's going to be development, then assay validation, and then regulatory approval. These are three processes. So when you do the assay validation, we are establishing three facts. One, the assay performance, we learn ourselves, and assay limitation. And third, of course, you have to get the regulatory approval to run the assay. So within that mind, in Acuities, we approach this problem entirely different. We look at the assay as an entirety rather than part by part. For example, in urinary tract infection, let us define first the matrix. Then we have to define how we collect the sample and transport. You have to produce a document, very clear document to regulatory agencies, how we are going to do it. Then the sample requisition form, how we are going to supply this to the clients. They want to see how it's going to look like. Then in the SOP, you have to have a clear definition for sample acceptance and rejection criteria. Then sample stability should be defined as well, which is we are going to validate when you do the analytical phase. Then based on the matrix, we have to define what are the interfering agents. Then extraction process, PCR, and results acceptance criteria. Then we have to submit a report. It's not just a single report, at least minimum 2 report, positive, negative, on top of that, based on our assay validation, any interfering agent, which is lower your LOD, or any cross-reactivity. You have to give additional example report, then clearly defined proficiency testing. Next slide, please. Next slide, please, Dr. Winslow. Thank you. Then we are preparing for the validation. When we prepare for the validation, we have to develop a validation plan. When you develop a validation plan, the first thing, of course, your testimonial. For that, you have to collect the literature. You have to do a literature survey to show the clinical relevance for the particular analyte. So you have to submit those literature to the regulatory agency. Then here, what we do normally, we do the theoretical LOD calculation. The reason is we want to take the assay performance in our hand rather than dictated by itself. At the same time, it also going to guide you if there is any problem in the validation, where to troubleshoot. That's where our success here. Next slide, please. When you prepare for the validation, the first thing, of course, matrix, the regulatory agency, they want to spike the material or use anything in the assay is a negative matrix. Here, the normal human pooled urine is commercially available. We also bought it for the commercially one, but we test it out on the culture plate as well as in the test system here in the UTA test menu. Make sure that everything is negative. Then we use the matrix for our experiment. So the regulatory agencies, they want to see the tighter intact organism. There is no synthetic DNA involved here. The only one case they are okay with that is a genomic DNA, for example, mycoplasma. It's very difficult to culture. At the time, they are okay with the genomic DNA. Other than that, they want to see all analytes in intact organism. And it is right now available from different sources. I listed few here. You can able to get. Next slide, please. So the validation plan should include specificity, include like inclusivity, exclusivity as well, and checkerboard, sensitivity, precision, assay interference agents, accuracy, and stability. These things should be included in the validation plan. Next slide, please. So on top of this validation, the first thing we focus on the extraction. We all know that not all microorganisms are not going to lyse equally. For example, gram-positive organisms are difficult to lyse. So to overcome the issue, we are going to usually optimize the extraction. For example, if you have a drug-resistant organism, gram-positive drug-resistant organism, in some cases, the cell wall is modified. So those organisms are not going to be lysed by lysozyme. In that case, we may have to consider like mutonolysine, lysostaphin, depends upon the organism. So we want to optimize first to make sure that we have the ability to lyse and isolate the nucleic acids. At the same time, we also focus on volume of beads if you are using any bead-based extraction and the protease gate. Of course, we pay attention in elution volume, which is going to dictate finally for the sensitivity of the assay. Then we focus on controls. Apart from NTC and NAC, we pay attention in detail for the spike in control. The regulatory agencies don't like to see one CT value and below. They want to see what is our acceptable CT range. We have to run the experiment and show this is your acceptable CT range, which is also applicable to your positive control as well. They want to, you have to define very clearly in our validation document. Next slide, please. So we always run the specificity experiment first, especially the inclusivity experiment. If you are using open array, it's an added advantage because it's a spatial multiplex. So if you are testing one organism, that means you are testing against 55 other different targets. So that's an added advantage here. On top of that, you have to select a few more organisms, genetically related organisms, commonly found in the matrix to demonstrate your assay performance is really good. Don't forget to include human DNA. The reason is there is not just a specificity, sometimes a human DNA can interfere in your assay. So one experiment can be reused in the assay interference agents. Next slide, please. So after you run the specificity experiment, then you move to the sensitivity. The sensitivity requirements are 6 to 10 point dilution series. Wait a minute. My test panel is too big. Usually UTI is going to be more than 30 organisms. That means there's a lot of testing. How we overcome this? So what we do is we make a pool of organisms. When we make the pool, we want to make sure that they're genetically distinct. Then you can run the sensitivity. This way, we can reduce the time and reduce the amount of money you spend to run this experiment. Usually, we combine LOD and precision together so that your validation progress really fast and save money. Next slide, please. So accuracy, there is a, do we need a clinical sample? No. Contrived sample is accepted. only if you spike those material in negative matrix. It should be spiked in at least 2 different concentrations, near and above, one above the LOD. They want to see minimum 30 samples per analyte. So on top of that, you need a 30 negative sample for the 2x2 table analysis. We collected some reference material from another laboratory used in the experiment. When you do those things, collect the report as well. De-identified report you need to further submission. Next slide, please. Then stability. The stability must be defined in 2 ways, one at a room temperature and at refrigeration. Plus, you need to establish the nucleic acid stability. What we did here, we did 4 times freeze the cycle, then we analyzed, and then we demonstrated the nucleic acids were stable. When finally you do the data analysis, we have to pay attention in detail. It should be analyte specific. And you have to present every single data in a table format in clear way. With that, I will show you one last slide which contains some clinical data. Next slide, please. So these are not reference material. These are actual clinical isolates. These are, we grow it in the lab and we took the live organism, we spiked in the urine, transferred to the collection device in 2 different concentrations and incubated at an ambient temperature and in cold in the refrigerated conditions. Then we lysed the cells, we ran the PCR assay. What is interesting I want to show you is at the higher concentration, over the period in ambient temperature, the CT value is going down. That's an indication of that this organism is growing. We all know that the urine is nutritious. They tend to grow. So why I'm showing this? The intact organism, live organism, when you include in the validation, you can understand the assay performance really well. Whether clinically this is going to affect the patient, the answer is no. The reason is most of the organisms are tend to grow when we spike in 100,000 CFE. But it will give you an idea what is going on in your assay performance. Plus, in using intact organism, you are defining not just the PCR, include the extraction as well. You are demonstrating you have the ability to lyse the organism and extract the nucleic acids. Thank you. Dr. Winslow, can you please take over?
Yes. So just to summarize my information and Dr. Mayil's information, I want to state that it is very important. UTI diagnostic methods absolutely matter in the clinic. So we have the patient, we have the healthcare provider. The healthcare provider, of course, is going to do a clinical evaluation. But the next step, choosing the right test for the right patient at the right time, what we know as diagnostic stewardship, is absolutely critical for the clinical outcomes that we will have on the other side of the diagnostic test. By choosing a rapid diagnostic test for urinary tract infection detection and having that rapid diagnostic result reported, as well as the right interpretation of the information, the right antimicrobial choices at the right time, will lead to those positive clinical outcomes that we want to see from the diagnostic method that we choose for urinary tract infection detection. So thank you for joining us today. We're going to now open the floor for questions.
Thank you, Ms. Winslow and Dr. Shanmugam, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until November 9th, 2024. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.